Good morning everybody. Students, today we see chapter 9, application of biotechnology. Yesterday we have studied the application of biotechnology in medicine. Today we continue the remaining concepts. 9.2 gene therapy, then the two strategies, two approaches and process of gene therapy. Followed by 9.3 stem cell therapy. We have two types, embryonic stem cell, then adult stem cell. Then 9.4 molecular diagnostics. So these are all the concepts today we are going to learn. Students, 9.2 gene therapy. What do you mean by gene therapy? Why it is required? Now you consider a person is infected by a disease. So what we do? So we go for taking the medicine and thereby we give the treatment. But in the case of a hereditary disease, in the case of a genetic disorder, what we can do? So here we have to treat the genes. So gene therapy is a technique that uses genes to treat or prevent the disease. So here in the picture itself, it is very clearly given what is the meaning of gene therapy. See here, the particular defective gene, when it is defective, when the gene is defective, we can replace it because it produces defective phenotype. Say for example here, we introduce the particular gene through a vector and thereby it produces normal phenotype. The expression of normal gene in the person results in a functional gene product whose action produces normal phenotype. So this is carried out through a vector. We can say the example from cystic fibrosis and hemophilia. So in these two, the main thrust of gene therapy has been directed at correcting single gene mutation. At present, most genetic diseases have no effective treatment and the gene therapy could often hope for many people. So here we have two strategies. One is gene augmentation therapy and another one is gene inhibition therapy. So what is the meaning of these two? In the first case, that is gene augmentation therapy, it involves insertion of DNA into the genome to replace the missing gene product. So here we correct it by inserting the DNA. But in the case of second one, gene inhibition therapy, it involves insertion of antisense gene and which inhibits the expression of the dominant gene. So here in the second one, we suppress the effect. But in the first one, we introduce the functioning gene and it expresses to produce the cell functioning normally. This is about the two strategies. And we have two approaches in gene therapy. One is somatic cell gene therapy and another one is germline gene therapy. The insertion of functional and expressible gene into a target somatic cell to correct a genetic disease is called somatic cell therapy. Then germline cell therapy, it is introduction of DNA into germ cells which is passed to the successive generation. So what is the meaning of this one? In the first case, somatic cell gene therapy, we insert the particular functioning gene to the body cell so that the genes can be expressed and rectify the genetic disease for the particular person only. But in the case of germline gene therapy, here the gene is introduced into the germ cells, say for example the eggs and the sperm, so that they are heritable and they are passed to the later generation. So when we compare somatic and germline gene therapy, we have different, then pass on to the process of gene therapy. Here students, we have three important stages in the process of gene therapy. One is isolation of a specific gene. Second one is making its copies and inserting them into target cells. Here the first clinical gene therapy was given in the year 1990 by French Anderson to a 4-year-old girl with adenosine deaminase deficiency. So what is ADA deficiency? So it is a adenosine deaminase deficiency which causes SIT. What is it? Severe combined immunodeficiency. It is an autosomal recessive metabolic disorder. It is caused by deletion or the duplication of the gene coding for ADA enzyme. In this person, the non-functioning of T lymphocytes cannot elicit immune responses against invading pathogens. The right approach for sick treatment would be to give the person a functioning ADA which breaks down toxic biological product, so that we follow the process and thereby we rectify the deficiency of ADA. So T lymphocyte, you know very well, it is very important in the immune process. That's why when there is a deficiency of T lymphocytes and it results in severe combined immunodeficiency disease. The genetically engineered lymphocytes. So first one, we take the gene of interest, that is in this case we take the normal human ADA gene, that is our gene of interest, and we want to introduce that one to clone, that is we take genetically disabled retrovirus. Then we incorporate the in particular to the virus, and there they are multiplied many number of cells, and now the culture is introduced into the particular person. This is called the process of gene therapy. Next, pass on to the stem cell therapy. The stem cell therapy, I hope you know very well, it is undifferentiated cell. What do you mean by undifferentiated cell? It can differentiate into any kind of cells, that is called undifferentiated cell. The stem cell has the ability to regenerate damaged and diseased organs. It is capable of self-renewal and exhibit cellular potency. The stem cell therapy can differentiate into all type of cells that are derived from any of the three germ layers. So the germ layers are ectoderm, endoderm and mesoderm. So before we pass on to the further, we need to understand the meaning for some technical terms. Totipotency, pluripotency, multipotency, oligopotency and unipotency. So students, you have to remember these words in the correct order. Then only you can easily know the meaning for that. Totipotency, that is total ability. So here the single cell can divide and produce all kinds of cells in the organism. That is called totipotency. Then pluripotency means, pluri means several. It refers to a stem cell that has the potential to differentiate into any of the three germ layers, that is ectoderm, endoderm and mesoderm. Multipotency, I hope you know the meaning for multi, that is many. It refers to the stem cells that can differentiate into various types of cells that are related to them. For example, blood stem cell can differentiate into lymphocytes, monocyte, neutrophils and other blood cells. Oligopotency, oligo means a few. It refers to stem cells that can differentiate into a few cell types only. For example, lymphocytes or myeloid stem cells can differentiate into B and T cells but not RBC. So here we have the limitation. Only a few cells can be differentiated. And unipotency, uni means single. It refers to ability of the stem cell to differentiate into only one type of cell. So students, here we have studied 5 different ability of the stem cells. Totipotency, pluripotency, multipotency, oligopotency and unipotency. Now, what are the 2 main types of the stem cells? We have embryonic stem cells and adult stem cells. Now we see one by one. First one is embryonic stem cells. It is derived from undifferentiated inner mass cells of a human embryo. It is a pluripotent and can produce the three primary germ layers, ectoderm, mesoderm and endoderm. It is isolated from the epiplast tissues of the inner cell mass of a blastocyst. When stimulated, the embryonic stem cells can develop into more than 200 cell types of the adult body. The ES cells are immortal. So these are all some basic characteristic feature of embryonic stem cells. First one, it is undifferentiated cell, pluripotent. Then it is isolated from the epiplast tissue of the inner cell mass of blastocyst. It is immortal. When you come to the adult stem cells, here it is found in various tissues of children as well as adult. So what is the difference? Here it is present, the embryonic stem cell is present in the embryo, but in the case of adult stem cell, it is present in the children or in the adult. That is a major difference. An adult stem cell or somatic stem cell can divide and create another cell similar to that. But in the case of embryonic stem cell, it is a pluripotent and it can differentiate into ectoderm, mesoderm and endoderm. And here it is a multipotent and embryonic stem cell is a pluripotent. The adult stem cells, we can say it can act as a repair system of the body. So whatever there is a repair record, the multipotent nature of the adult stem cell can rectify the problem. The red bone marrow is a rich source of adult stem cell. Then what are the applications of stem cells? Whether it is a embryonic or adult stem cell, what are the applications? First one is generation of cells and tissues for the cell-based therapies. So we can go for the therapy based on the generation of the particular cells. Then to test new drugs. So for these two processes, we need the stem cell. Now we have a new banking system, that is stem cell banking. What is stem cell banking? Stem cell banking is the extraction, processing and storage of stem cells so that they may be used for the treatment in the future when required. The banking, when you store the particular stem cell, they cost and we have to pay it. Like we store the blood, here we can store the stem cells. That is called stem cell banking. We can use it when there is a need. That is a meaning for stem cell banking. Now we have different types of cell banking. One is amniotic cell banking. It is a facility that stores stem cell derived from amniotic fluid for future use. So see here, we take the amniotic fluid. So the amniotic fluid is stored for future use. And next one, cord, umbilical cord, blood banking. So the picture clearly shows that one. It is the extraction of stem cell from the umbilical cord during childbirth. While the umbilical cord and cord blood, it is nothing but the blood present inside the umbilical cord. See here, in this you can observe the cord blood are the most popular sources of stem cells. The placenta, amniotic sac and amniotic fluid are also rich sources in terms of both quantity and quality of stem cells. In all these we can store the stem cells. 9.4 Molecular diagnostics. So students, now you assume there is a disease. How do you know there is a disease? So the disease you can easily identify through the symptoms. When there is a symptom, that means already there is a manifestation of the disease. So before the symptom occurs, we have to start the treatment. So for that we need the molecular diagnostics. So it is for the appropriate treatment, but molecular diagnostic is not a treatment method. It is a diagnostic method. You can early detect the particular disease. That is a meaning for molecular diagnostics. These laboratory techniques are indirect and not always specific. Say for example, the presence of pathogen like virus, bacteria is detected only when the pathogen produces symptom in the person. By the time the symptom appear, concentration of pathogen becomes very high in the body. So it is very difficult to treat in the later stage. However, very low concentration of bacteria or a virus, even when the symptoms of the disease does not appear, can be detected by amplification of the nucleic acid. So this is the main aim of molecular diagnostics. So even when there is no symptom, we can amplify the nucleic acid of the virus or bacteria, and thereby we can start the treatment earlier. The techniques that are reliable and help in early diagnostics are recombinant DNA technology, polymerase chain reaction, and enzyme-linked immunosorbent assay. So first one, rDNA technology, second one is PCR, and third one is ELISA. So these are all the three techniques we are going to study in this concept. First one is enzyme-linked immunosorbent assay. This was discovered by Eva Enkwal and Peter Perumal in the year 1971. So what is the aim of this ELISA technique? To detect the presence of specific antibodies or antigen in a sample of serum and urine. So when the doctors collect the serum or urine, so they can easily identify the particular antibodies or antigen by ELISA method to detect if a person is HIV positive or negative. When you study ELISA, it is more related to the second point, that is to determine if a person is HIV positive or negative. And third point is to determine serum antibody concentration, that is how much a person can produce the particular antibodies when the HIV pathogen enters inside. Then fourth point is to detect the presence of specific antigen and hormones such as human chorionic gonadotrophins. So these can be detected with the help of ELISA. Here this method is highly sensitive and specific. The ELISA possesses the added advantages of not requiring radioisotopes or a radiation counting apparatus. Nothing required. No sophisticated instruments are required to conduct ELISA technique. The process of ELISA. Now, during the diagnosis of sample, the sample is immobilized on the surface of an ELISA plate. Then the antibody is added and it is allowed to react with the immobilized antigen. Now it is linked to an appropriate enzyme like peroxidase. The untreated antibody is washed away and the substrate of the enzyme, that is hydrogen peroxidase, is added with certain reagents such as chloronaphthol. Then the activity of the enzyme yields a colored product indicating the presence of antigen. The intensity of the color is directly proportional to the amount of antigen. So by this way we detect the presence of antigen in the particular sample. This is the diagram given in your book. So here in the two diagrams, this is the diagram given in your book and this is a reference. Okay, in this power you can easily identify the four stages. One is, first one, coating. The antigen is adsorbed. So adsorption, you know very well, it is attached above the surface of the wall. And next one, it is blocking. A buffer containing unrelated protein is used to block free sites in the wall. So blocking. Then third stage is detection. The enzyme conjugated detection, antibody binds antigen. Then fourth one is readout. Here the substrate is catalyzed by the enzyme to generate colored readout. So from this we can easily find out the presence of the particular antigen. We have four types of ELISA: direct ELISA, indirect ELISA, sandwich and competitive ELISA. So these are all the pictures to explain, to understand the particular types of ELISA. Direct. So in the first one, direct ELISA, the substrate is directly linked, and indirect ELISA, there is secondary antibody conjugate through which the substrate is attached. In the third one, sandwich ELISA, we have capture antibody and there is a substrate later. In the competitive ELISA, we have inhibitor antigen also. So it has to compete with that later. We identify the particular one. So these are all the four kinds of ELISA. Now PCR, polymerase chain reaction. What is PCR? Polymerase chain reaction. Students, it is nothing but very simplest one. How we take Xerox copies. You know very well. You have the handouts, so many notes. So from that you can take n number of Xerox copies. So like that, PCR is an in vitro amplification technique used for synthesizing multiple identical copies of DNA. So like Xerox copies, here we take the copies of DNA. That's all. In vitro amplification technique. It is developed by KaryB Mullis. He was a Nobel laureate in the year 1993. The entire process has three steps. One is denaturation, renaturation or primer annealing, and third one is synthesis or primer extension. Now the entire process we can call the thermal cycler. The thermal cycler is nothing but it is the place where the three stages are taking place. We can compare the thermal cycler like the Xerox copier, the photocopier. So in that the n number of DNA copies are produced. Children, this is called the cycler, thermal cycler. The double-stranded DNA of interest is denatured to separate into two identical strands by high temperature. This is called denaturation. Each strand is allowed to hybridize with a primer renaturation or primer annealing. The primer template is used to synthesize DNA by using Taq DNA polymerase. During denaturation of reaction, mixture is heated to 95 degrees Celsius for a short time to denature the target DNA into single strand that will act as a template for DNA synthesis. Annealing is done by rapid cooling of the mixture, allowing the primer to bind to the sequence on each of the two strands. During primer extension or synthesis, the temperature of the mixture is increased to 75 degrees Celsius for the sufficient period of time to allow Taq DNA polymerase to extend each primer by copying the single strand template. At the end of the incubation, both single template DNA strand will be made partially double strand. The new strand of each double-stranded DNA extend to a various distance downstream. These steps are repeated again and again to generate multiple forms of desired DNA. This process is also called DNA amplification. See here the cycle speed, how many copies are produced also. Within 4 hours, we can produce million numbers of DNA copies. So students, here we have three processes. One is denaturation, renaturation or primer annealing. What we have observed in the video is given in the sentence form. And here you must know what is the meaning of Taq DNA polymerase. You know very well the polymerase enzymes are made up of protein. Protein when heated to 95 degrees Celsius, definitely they denature. But we have a specific DNA polymerase from the bacterium called Thermus aquaticus. So from that we have the DNA polymerase. So that can withstand 95 degrees Celsius. Then we have the primer extension or synthesis. So when you see in the picture, you can observe the extension of the DNA takes place from 3' end to 5' end in both template. Very specific one, that's why we call it is a amplification technique. Amplification of the particular DNA. Next, PCR in clinical diagnosis. Diagnosis of inherited disorders. Say for example, the genetic disease, viral disease or bacterial disease can be diagnosed. PCR is also employed in the prenatal diagnosis of inherited diseases by using chorionic village sample or cells from amniocentesis. Sickle cell anemia, beta thalassemia, phenylketonuria can be detected with the help of PCR. cDNA from PCR is a valuable tool for diagnosis and monitoring retroviral infections. Say for example, tuberculosis by Mycobacterium tuberculosis. Cervical cancer caused by Polymerase can be detected. And fifth one is to detect sex-linked disorders in fertilized embryos can be detected. I hope students nowadays the RT-PCR. So what is RT-PCR nowadays? You come to know this word in the coronavirus diagnosis. RT. So this is for DNA viruses. So in DNA viruses, the DNA is heated and it is made to template. So from the template, the new DNAs are produced. That is the main concept in the first one. But in RT, the reverse transcriptase is converting the RNA into DNA. So for that we need the reverse transcriptase. That's why we called for the coronavirus detection, we call RT-PCR. RT-PCR. Why? Because here the virus has RNA. So for RNA viruses, we have to go for RT-PCR. Then application of PCR, very important in the study of evolutions, more specifically phylogenetics. So what is the meaning of phylogenetics? It is a systematic study of organism relationship based on evolutionary similarities and differences. It can be used to amplify even minute quantities of DNA from any source. Say for example, hair, mummified tissues, bones or any fossilized materials. It is used in the field of forensic medicine. PCR is very useful for identification of criminals. And last point is PCR is also used in amplification of specific DNA segment to be used in gene therapy. So I have given the color. Second point and fifth point, these two are looking somewhat same. That is a amplification. It can be amplified and they are used for gene therapy. And third and fourth, it is used to find out the criminals, or we can say in the forensic medicine it is very helpful. And first one, we can use it for phylogenetic studies. So these are all the 5 application of PCR. And next one, transgenic animals, it is deleted part. how we can produce, then use of transgenesis, that is also deleted. Biological product and their uses, that is also deleted portion. Animal cloning, advantages of animal cloning, then disadvantages of animal cloning. Thank you children. Today we have studied about the gene therapy and two types of gene therapy, stem cell therapy. Then we have studied about the molecular diagnosis. In that PCR is very important, and the application is sometime they may ask for 5 more questions. Thank you children. We meet again in another lesson. Thank you.